Hi, I'm Antonio Clemente, a master student in immunology, and together with my advisor Jessica Meiro, PhD, we are developing the work intercellular communication mediated by adipose tissue-derived extracellular vesicles, immunological impact on PBMCs and NK cells. To begin, obesity is not just an excess of body fat. Adipose tissue work as an active endocrine and immune organ capable of secreting hormones, adipokines, and modulating immune responses. In obesity, adipose tissues expands, becomes infiltrated by immune cells, and maintain a state of chronic low-grade inflammation. A key population affected in this environment is natural killer NK cells, whose cytotoxic function can become impaired despite inflammatory activation. This raises interest in understanding whether extracellular vesicles (EVs) released by adipose tissue contribute to altering NK cell activity. So, even after weight loss, many individuals maintain inflammatory signatures, suggesting an immune memory of this obese state. Because adipose tissue release extracellular vesicles contain proteins, lipids, and RNA cargo, this vesicle may influence NK cell activation and cytotoxicity. Our hypothesis is that adipose-derived EVs modulate NK cell behavior, particularly their ability to respond to tumor target through missing self-recognition. So, the general objective of this project is to investigate how adipose tissue-derived EVs interact with NK cells and how these interactions shape their activation and cytotoxic responses. Furthermore, the project includes three main objectives like evaluate how EVs modulation activation, exhaustion markers, cytokine production, and viability in PBMC derived NK cells, assess NK cell functional markers such as granzyme B, perforin, and CD107A, as well as their proliferation, and examine whether EVs after NK cell and tumor cytotoxicity using tumor cell lines that differ in HLA-1 expression like DAUDI, K562, and MCF7, allowing assessment of missing self-driven activation. So for our methods, EVs will be isolated from adipose tissue of obese and eutrophic individuals using differential centrifugation and ultracentrifugation. Characterization will include NTA, DLS, and electron microscopy. PBMCs will be exposed to EVs and then analyzed by flow cytometry for activation markers, exhaustion markers, and cytotoxic signatures in NK cells. Proliferation will be assessed using CFSE dilution. Finally, NK cells will be co-cultured with tumor cell lines like DODI, K562, and MCF7. These targets vary in HLA-1 expression and allow us to test how EVs affect missing cell recognition and NK mediating killing. We expect that adipose-derived EVs from obese and eutrophic donors to show distinctive biological profiles that differentially modulate NK cell activation and cytotoxicity, include altered killing of HLA-1 low and HLA-1 variable tumor cells. These findings may provide insight into NK dysfunction in obesity and highlight EVs as potential immunomodulatory tools with relevance for cancer immunosurveillance in metabolic disease. Thank you for attention. This is our reference. Goodbye.